Imagine, for a moment, the incredible unseen operations happening inside you right now.
Yeah.
Your body's taking something as fundamental as a sugar molecule, our primary energy currency, and in this really intricate molecular dance, it's transforming it. Sometimes into immediate energy, maybe to power your thoughts, your movements, other times into brand new building blocks to create and repair cells, or even into these specialized protective agents.
And it's not just happening once in a while, it's constant.
Exactly. A precisely managed economic system playing out billions of times a second. It's kind of mind-blowing when you think about it.
Yeah.
Welcome to the Deep Dive. I'm your guide into the fascinating world of knowledge, and joining me today, as always, is our brilliant expert.
It's great to be here. Yeah, today we're really embarking on a foundational exploration right into the heart of how cells manage their energy and create essential molecules.
Absolutely. Today we're extracting the most vital insights from a really crucial chapter of Lehninger Principles of Biochemistry. That's the 8th edition by Nelson and Cox, specifically Chapter 14.
Right, the one focusing on glycolysis, gluconeogenesis, and the pentose phosphate pathway. Core stuff.
Exactly. Think of this as a comprehensive, sort of pedagogically structured summary. It's perfect if you're curious about the molecular mechanisms underpinning life, or maybe if you're an upper division undergrad looking to clarify these sometimes complex processes.
Yeah, our mission is basically to take these core biochemical pathways, the enzymes involved, the energy dynamics, how it all fits together.
Metabolic integration.
Precisely.
Oh, yeah.
And translate it all into accessible language, helping you understand how life really functions at that molecular level.
So if you've ever wondered how your body fuels its every move, or how it manages energy when supplies run low, or even how it fights off cellular damage, then you're definitely in the right place. Okay, let's dig in. Let's kick off with glycolysis. The name itself comes from Greek, meaning sweet splitting, doesn't it?
It does, and it's this nearly universal metabolic pathway. It's really the body's primary way to start breaking down glucose.
And it has quite a history, too.
Oh, absolutely. Its historical significance is huge. Glycolysis was actually the very first metabolic pathway to be fully understood. It all started back in 1897 with Edward Buckner.
Ah, the yeast extracts.
Exactly. His discovery that fermentation, a life process, could happen in yeast extracts outside living cells.
Yeah.
That was revolutionary.
It wasn't just science. It was like a philosophical shift.
Totally. It showed life's reactions could be explained chemically. And that just, opened the floodgates for biochemical research enzyme purification, discovering things like NAD plus ions, the role of ATP, foundational stuff.
So this initial breakdown, glycolysis.
Yeah.
What does it mean for us, for our cells?
Well, it's incredibly central. I mean, for many cells, think red blood cells, the brain, even some plant tissues, glycolysis is their only source of metabolic energy.
The sole source, wow.
Yeah, and the chemistry itself has been perfectly conserved through evolution. The enzymes in us are remarkably similar to the ones in yeast or spinach. It's like a universal language of energy, proven efficient over millennia.
Okay, so the pathway itself, it happens in 2 main stages, right? The preparatory phase and the payoff phase.
That's right. Investment first, then the returns.
So tell us about that investment part, the preparatory phase.
Okay, so in this first phase, the body makes a clever investment. It actually spends 2 ATP molecules.
Spends energy to start.
Exactly. It uses them to tag the glucose molecule through phosphorylation. This does 2 key things. 1, it makes the glucose more reactive, ready for splitting. 2, it traps the glucose inside the cell.
Traps it. How?
Because phosphorylated sugars can't easily cross the cell membrane. There are no transporters for them. So once it's tagged, it's committed.
Oh, smart.
Then comes the actual sweet splitting, the lysis step. That tagged 6-carbon sugar gets cleaved right in half into 2 smaller 3-carbon molecules.
And those 2 are then ready for the next stage.
Precisely. Ready for the payoff phase.
Okay, so that initial investment makes sense. Trapping the glucose, getting it ready. What happens in the payoff phase? How do we start getting energy back?
Right, so now those 2 3-carbon molecules get transformed. There's a critical energy-conserving step where they're oxidized, and inorganic phosphate, not from ATP this time, just free phosphate is added directly.
Okay.
This creates a high-energy compound. And here's the cool part. In a process called substrate-level phosphorylation, this high-energy compound directly donates its phosphate group to ADP, making ATP.
Directly. No complex machinery needed.
Nope. Straight from the substrate to ADP. And this happens twice, once for each of those 3 carbon molecules.
So we get 2 ATP back there. We've broken even on the initial investment.
Exactly. Then, through a few more rearrangements, another high-energy compound is formed called PE-phosphenol pyruvate.
Right.
And in the final step, PEP donates its phosphate to ADP, generating more ATP and leaving us with the final product, pyruvate.
So 2 more ATPs, one from each 3-carbon path.
Correct.
Okay, so if we look at the whole process, the overall balance sheet for glycolysis, what's the net gain from splitting one glucose?
For each glucose molecule that goes in, the net yield is 2 molecules of ATP.
Okay, so 4 produced minus the 2 invested. Net 2 ATP.
Exactly. And you also get 2 molecules of NADH.
NADH.
Yeah.
That's like captured energy, right? An electron carrier.
Precisely. It holds high-energy electrons ready to be cashed in later for much more ATP, but only if oxygen is available.
And the whole process, glycolysis itself.
It's energetically favorable. Overall, it has a large negative free energy change, making it essentially irreversible in the cell. It's driven strongly towards completion.
So 2 net ATP and 2 NADH.
Yeah.
But is that all the energy we can possibly get from that starting glucose molecule? It feels like there should be more locked away in that pyruvate.
You're absolutely right. Glycolysis only extracts a small fraction of glucose's total potential energy. Those 2 pyruvate molecules are still energy-rich.
So what happens to them?
Well, that depends. Under aerobic conditions, when oxygen is plentiful, pyruvate gets shipped into the mitochondria.
The cell's powerhouses.
Right. And there, it enters the citric acid cycle and goes through oxidative phosphorylation. That's where the vast majority of ATP is generated. We're talking maybe 32 ATP per glucose in total.
Ah, okay, that's the big payoff.
That's the big payoff. But under anaerobic conditions, when oxygen is scarce, pyruvate takes different routes we call fermentation.
Which we'll get to, but first, glucose is the main player. But it's not the only sugar that can fuel this, is it?
No, not at all. That's a great point. Cells are incredibly efficient, really economical. They don't bother having completely separate, complex pathways for every single sugar they might encounter.
So they funnel others into glycolysis.
Exactly. They convert various carbohydrates into existing glycolytic intermediates. Think of it like on-ramps to the main highway.
Oh, what kind of sugars?
Well, take our own energy stores, glycogen in animals, or starch in plants. When these are broken down, they don't just release free glucose. They use a process called phosphorylasis.
Phosphorylasis, not hydrolysis.
Right. It uses phosphate to break the bonds, yielding glucose 1-phosphate, which is quickly converted to glucose 6-phosphate.
Which is already the first intermediate in glycolysis after the initial ATP investment.
Precisely. So it bypasses that first ATP-consuming step. That actually gives the cell a net gain of 3 ATP per glucose unit from glycogen instead of just 2 from free glucose. It's a neat energy-saving trick.
Clever. And what about sugars we eat?
Most dietary carbs, like starch or sucrose, are first broken down by digestive enzymes, amylase, sucrase, etc., into simple sugars, monosaccharides, glucose, fructose, galactose. Only those single units get absorbed into the bloodstream and then into cells.
This actually brings us to something many people experience: lactose intolerance.
Ah, yes, a very common example. Lactose, the sugar in milk, is a disaccharide. It needs to be broken down by an enzyme called lactase into glucose and galactose to be absorbed.
But many adults lose that enzyme.
Exactly. Especially populations outside of Northern Europe, many people experience a natural decline in lactase production after childhood. Without enough lactase, the lactose passes undigested into the large intestine.
And then the gut bacteria have a field day.
Pretty much. They ferment the lactose, producing gas, acids, and other products that cause those uncomfortable symptoms: cramps, bloating, diarrhea. It's a classic example of a common genetic variation impacting diet.
So what about those other single sugars, like galactose from milk or fructose from fruits and honey? How do they get onto the glycolysis highway?
They each have specific entry points. Galactose, for instance, is phosphorylated and then converted through a series of fairly complex steps involving UDP glucose into glucose 1-phosphate and then glucose 6-phosphate, ready for glycolysis.
And if those steps go wrong?
Defects in those galactose-converting enzymes cause conditions called galactosemias. These can range from relatively mild issues like cataracts to much more severe developmental problems and liver damage if not managed early through diet.
And fructose.
Fructose metabolism is interesting because it differs a bit depending on the tissue. In muscle and kidney, it can be directly phosphorylated by hexokinase to fructose 6-phosphate, a glycolytic intermediate.
Simple enough.
But in the liver, it takes a different route. It's phosphorylated to fructose 1-phosphate, then cleaved into 2,3-carbon fragments. Both of those fragments are then converted into glyceraldehyde 3-phosphate, another key glycolytic intermediate.
So it bypasses some of the early regulatory steps.
It does, and that has some implications for metabolism, particularly with high fructose intake, which we might discuss another time. But essentially, it feeds right in. Mannose, another sugar, is handled similarly phosphorylated, then isomerized into fructose 6-phosphate.
Okay, so after all that sweet splitting, whether starting from glucose or another sugar, we end up with pyruvate. And you said pyruvate is at a crossroads.
A major metabolic crossroads. Its fate really depends on the cell's needs and, crucially, on the availability of oxygen.
3 main fates you mentioned.
3 main catabolic fates, yes. We already touched on the first, under aerobic conditions, with plenty of oxygen.
Into the mitochondria for the citric acid cycle and oxidative phosphorylation. Big ATP payoff.
Exactly. Complete oxidation to CO2 and water, regenerating lots of NADH and FADH2, which fuel massive ATP synthesis via the electron transport chain.
Okay, fate number 1. What about when oxygen is limited? Anaerobic conditions or hypoxia?
Right. If oxygen is scarce, the cell can't run the electron transport chain efficiently. But glycolysis needs a steady supply of NAD+ to keep going, specifically for that oxidation step in the payoff phase.
Ah, the NADH produced earlier needs to be recycled back to NAD+ somehow.
Precisely. Without oxygen to ultimately accept those electrons via the respiratory chain, the cell needs an alternative way to regenerate NAD+ COM, and that's where fermentation comes in. It's all about NAD+ regeneration so glycolysis can continue.
Got it. So what are the fermentation options?
2 main types are relevant here. First, lactic acid fermentation. This happens in animal tissues, like our muscles during intense, short bursts of exercise when oxygen supply can't keep up with demand, or in cells that lack mitochondria entirely, like red blood cells.
What happens?
Pyruvate itself acts as the electron acceptor. It takes the electrons back from NADH, reducing pyruvate to lactate and, crucially, regenerating NAD plus Nothi cells.
So glycolysis can keep making that little bit of ATP.
Exactly. It allows for those short bursts of strenuous activity. The lactate produced isn't just waste either. It can be transported to the liver and converted back to glucose later via the Cori cycle.
Okay, that's lactic acid fermentation. What's the other main type?
The other is ethanol fermentation, which is famous because of yeast, but also occurs in some other microorganisms and even plants under certain conditions.
Like in bread making or brewing.
Exactly. Here, it's a 2-step process. First, pyruvate is decarboxylated. A CO2 molecule is removed to form acetaldehyde. This step requires a crucial coenzyme called thiamine pyrophosphate, or TPP.
TPP, derived from vitamin B1.
Thiamine. That's the one. Then, in the second step, acetaldehyde accepts electrons from NADH, reducing it to ethanol and regenerating NAD plus West.
So different end product, ethanol instead of lactate, but the same goal. Regenerate NAD plus for glycolysis.
Precisely.
This regeneration need explains the Pasteur effect, doesn't it?
Yeah.
Where yeast consume way more glucose without oxygen.
It does. Louis Pasteur noticed yeast chew through glucose much faster anaerobically, and it makes perfect sense. Glycolysis alone gives only 2 net ATP per glucose. Aerobic respiration gives around 32.
Huge difference.
Massive. So to get the same amount of energy anaerobically, the cell has to process about 15 times more glucose compared to when oxygen is present.
And related to this, there's the Warburg effect. You mentioned it has implications for cancer.
Yes, this is a really fascinating and still intensely studied phenomenon. Otto Warburg observed back in the 1920s that many tumor cells exhibit surprisingly high rates of glycolysis followed by lactic acid fermentation, even when oxygen is available.
Even with oxygen, that seems wasteful, energetically speaking. They're choosing the 2 ATP route over the 30-plus ATP route.
It does seem counterintuitive, doesn't it? It's often called aerobic glycolysis. Why would rapidly growing cancer cells rely on such an inefficient process?
Yeah, why?
Well, the thinking is multifaceted. Part of it might be due to initially poor blood supply and hypoxic conditions within a growing tumor. Also, cancer cells often upregulate glucose transporters, grabbing more glucose. But crucially, while inefficient for ATP yield, rapid glycolysis provides something else vital for fast-growing cells: metabolic intermediates.
Building blocks.
Exactly. Carbon skeletons needed to synthesize lipids, nucleotides, amino acids, all the components needed for rapid cell division. Glycolysis, even if inefficient for ATP, might be faster at providing these precursors than complete oxidative phosphorylation.
So it's a trade-off. Less energy per glucose, but faster supply of building material.
That seems to be a major part of it, and what's really striking is how this metabolic quirk is used diagnostically.
The PET scans you mentioned.
Right. Positron emission tomography often uses a glucose analog, FDG, which is radioactively labeled. Cancer cells, with their high glucose uptake and glycolysis rates, avidly take up FDG, but they can't fully metabolize it past the initial steps.
So it accumulates.
It accumulates, and the radioactive signal allows doctors to pinpoint the location and activity of tumors. It's using the cancer cell's own metabolic signature against it.
And that TPP coenzyme needed for ethanol fermentation, you said it's vitamin B1.
Right.
That's essential for us too, right? Even though we don't ferment ethanol.
Absolutely critical. Thiamine pyrophosphate isn't just for yeast pyruvate decarboxylase. It's a vital cofactor for several key enzymes in human metabolism too, including pyruvate dehydrogenase, which links glycolysis to the citric acid cycle, and an enzyme in the pentose phosphate pathway we'll discuss later.
So a deficiency would cause problems.
Severe problems. Thiamine deficiency leads to conditions like Beriberi or Wernicke-Korsakoff syndrome, often characterized by neurological issues, because energy metabolism in the brain and nerves is heavily impacted. It really underscores how interconnected these pathways are.
It's amazing how fundamental these processes are, not just biologically, but how they've been harnessed culturally and industrially.
Oh, definitely. Fermentations are ancient technology. Think about food preservation and production. Yogurt, cheese, sauerkraut, kimchi, soy sauce, even the holes in Swiss cheese are due to CO2 produced during propionic acid fermentation.
And beverages, obviously. Beer, wine, champagne.
Of course. And beyond food and drink, industrial fermentations are used to produce biofuels like ethanol from corn or industrial chemicals like butanol and acetone. These microbes are essentially self-replicating little chemical factories.
Okay, so we've thoroughly covered breaking glucose down, glycolysis, and the fates of pyruvate. But what about the flip side? What if the body needs to make glucose, say, between meals or during prolonged fasting or intense exercise when glycogen stores run low?
That's where gluconeogenesis comes in. Literally means new formation of sugar.
Making glucose from scratch.
Pretty much. Synthesizing glucose from non-carbohydrate precursors, things like lactate, recycling it from muscle, pyruvate, certain amino acids derived from protein breakdown, and even the glycerol backbone from fats.
And where does this happen mainly?
Primarily in the liver. The liver is the main glucose producer for the rest of the body. The kidneys also contribute, especially during prolonged fasting, and a little bit happens in the small intestine.
And the glucose made is then released into the blood.
Exactly. To supply tissues that absolutely depend on glucose, like the brain, which uses a huge amount daily, and red blood cells, which have no other fuel source. And as we mentioned, it's key in the Cori cycle, turning that muscle lactate back into useful glucose.
Now, is gluconeogenesis just glycolysis running in reverse, like flipping a switch?
Ah, if only it were that simple. It shares many steps, but no, it's not just the reverse. Remember those 3 steps in glycolysis that were essentially irreversible due to large energy changes?
Right, the ones catalyzed by hexokinase, phosphofructokinase 1, and pyruvate kinase.
Exactly. Gluconeogenesis has to bypass those specific steps using different enzymes and different reactions. These bypass reactions are also energetically favorable, but in the direction of glucose synthesis.
Why the bypasses?
Yeah.
Why not just reverse the glycolytic enzymes?
Because simply reversing highly favorable reactions would require a huge energy input and wouldn't happen spontaneously. More importantly, having separate enzymes for these key steps allows for independent regulation.
So you can control whether you're breaking down or building up glucose.
Precisely. It prevents what's called a futile cycle, where both pathways run simultaneously at hot rates, consuming ATP and just generating heat with no net product. Separate control points are essential for metabolic efficiency.
Okay, let's look at those bypasses. The first one, getting from pyruvate back to phosphenolpyruvate, PEP, must be tricky, considering the pyruvate kinase step in glycolysis is so favorable.
It is tricky, and it's actually a 2-step process that involves both the mitochondria and the cytosol. It's quite elegant, really.
How does it work?
First, pyruvate enters the mitochondrion. There, an enzyme called pyruvate carboxylase adds a carboxyl group to it, forming oxaloacetate. This step requires biotin and consumes an ATP.
Okay, pyruvate to oxaloacetate inside the mitochondria.
Right. Now, oxaloacetate needs to get out into the cytosol, but there isn't a good transporter for it. So it's usually temporarily converted to malate, which can leave the mitochondrium.
Sneaky.
Once in the cytosol, malate is converted back to oxaloacetate. And then a second enzyme, phosphenylpyruvate carboxykinase, PPCK, converts oxaloacetate to P. This step uses GTP, another high-energy phosphate molecule, and releases the CO2 that was added earlier.
Wow, quite a detour. And costly. An ADP and a GDP used just to bypass that one glycolytic step for each pyruvate.
Exactly. 2 high-energy phosphate bonds per pyruvate, so 4 per glucose molecule synthesized. It underscores how important making glucose is. The body is willing to pay this high-energy price. There's also a variation if lactate is the starting material, where some steps can happen differently to manage electron carriers, but the core bypass strategy is the same.
Okay, that's bypass 1. What about the other 2 irreversible steps from glycolysis?
Those bypasses are simpler. To get past the phosphofructokinase 1 step of glycolysis, gluconeogenesis uses a different enzyme. Fructose 1-6-bisphosphatase, FBPase1, it simply removes a phosphate group from fructose 1-6-bisphosphate using water, releasing inorganic phosphate.
So not transferring to make ATP, just cutting it off.
Correct. It's a hydrolysis reaction. And the final bypass, getting past the hexokinase glucokinase step, involves the enzyme glucose 6-phosphatase.
And this one removed the last phosphate to make free glucose.
Exactly. It hydrolyzes glucose 6-phosphate to glucose and inorganic phosphate. Now, this enzyme is special because it's primarily found only in the liver, kidneys, and intestinal, lining the tissues responsible for releasing glucose into the bloodstream.
Ah, so other tissues like muscle and brain lack this enzyme.
Right. This ensures that the glucose 6-phosphate they produce or take up stays within those cells for their own energy needs. They can't release free glucose. It's a key part of tissue specialization.
So making glucose via gluconeogenesis is clearly energetically expensive.
Very. To make 1 molecule of glucose from 2 molecules of pyruvate, the net cost is 4 ATP, 2 GTP, and 2 NADH. That's a substantial investment.
But necessary, presumably.
Absolutely necessary. This high cost ensures the pathway runs effectively in the direction of synthesis when needed and allows for that crucial reciprocal regulation with glycolysis.
Quick question we touched on earlier. Mammals can't make glucose from fatty acids, right? Why is that again?
Right. No net synthesis of glucose from the typical breakdown products of most fatty acids. Fatty acids are mostly broken down into 2 carbon units of acetyl-CoA.
Okay.
In mammals, the reaction converting pyruvate, 3 carbons, to acetyl-CoA, 2 carbons plus CO2, is irreversible. There's no pathway to go backwards from acetyl-CoA to pyruvate or oxaloacetate, so those acetyl-CoA carbons can enter the citric acid cycle to generate energy, but they can't be used to build up a glucose molecule.
So the carbons are lost as CO2 in the cycle.
Essentially, yes, for the purposes of net glucose synthesis. However, the glycerol backbone of triglycerides can be converted to a glycolytic intermediate and then into glucose. So a small part of fat can become glucose.
But plants can do it.
Plants and many microorganisms have an extra trick, the glyoxylate cycle. It allows them to bypass the CO2-losing steps of the citric acid cycle and convert acetyl-CoA into oxaloacetate, which can then be used for gluconeogenesis. That's vital for seeds germinating, using stored oils as their energy and carbon source.
Okay, so we have these 2 major pathways, glycolysis, breaking glucose down, gluconeogenesis, building it up. They run in opposite directions, share many steps, but have these key irreversible bypasses. And you mentioned regulation is crucial to avoid them running full tilt simultaneously.
Absolutely essential. That would be incredibly wasteful, like flooring the accelerator and the brake at the same time, just burning ATP and generating heat for no reason. So they are tightly and reciprocally regulated.
Meaning when one is active, the other is generally suppressed.
Exactly. And the main control points are, unsurprisingly, those irreversible enzymatic steps, the bypass steps we just talked about.
How does the body manage this coordination? What are the signals?
There are multiple layers of control acting over different time scales. One immediate level involves the enzymes themselves. Take hexokinase, the first enzyme in glycolysis. There are different versions, or isozymes.
Like the ones in muscle versus liver.
Precisely. Muscle hexokinases, I3, have a high affinity for glucose. They grab it quickly even at low concentrations and are inhibited by their own product, glucose 6-phosphate, perfect for muscle needing immediate fuel.
Makes sense.
But the liver isozyme, hexokinase the 4th, or glucokinase, has a lower affinity. It only gets really active when glucose levels are high, like after a meal. And importantly, it's not directly inhibited by glucose 6-phosphate. Instead, its activity is regulated differently, involving movement in and out of the nucleus based on glucose levels.
So the liver acts like a buffer, only taking up significant glucose when there's plenty around.
Exactly. It prevents the liver from hogging glucose when other tissues might need it more urgently.
Okay, that's the first step. What about the other key control points? The PFK1 and FB-PACE1 pair sounds critical.
It's arguably the most important control point. Phosphofructokinase 1, PFK1, drives glycolysis forward, while fructose 156-bisphosphatase, FBPase1, drives gluconeogenesis. They are reciprocally controlled by the cell's energy state.
How so?
High levels of ATP, signaling energy abundance, inhibit PFK1, slowing glycolysis. Citrate, an intermediate from the citric acid cycle, which also indicates plenty of fuel from fat or protein breakdown, also inhibits PFK1.
So signals of enough energy shut down glucose breakdown.
Right. Conversely, high levels of ADP and especially AMP, which signal low energy charge, strongly activate PFK1, boosting glycolysis.
And FB-PACE1, the gluconeogenesis enzyme.
It's inhibited by AMP, so when energy is low, high AMP, the body stimulates glucose breakdown, glycolysis, and inhibits glucose synthesis. Gluconeogenesis makes perfect sense.
Then there's this molecule, fructose 2006-phosphate, F2006BP. You mentioned it's a master regulator.
Yes, F2006BP is a hugely important allosteric effector. It doesn't appear in the main pathways themselves, but acts as a signal. It potently activates PFK1, pushing glycolysis forward, and strongly inhibits FD-PACE1, slamming the brakes on gluconeogenesis.
So it's like a switch that flips metabolism towards glucose breakdown.
Exactly. And its own levels are controlled by a fascinating bifunctional enzyme 1 protein with two different activities, PFK2, which makes F2006BP, and FB-PACE2, which breaks it down.
Two enzymes in one.
Yep. And the balance between these two activities is controlled by hormones. Glucagon, released when blood glucose is low, triggers a modification phosphorylation of this bifunctional enzyme. This activates the FB-PACE2 part and inhibits the PFK2 part.
So glucagon lowers F2006BP levels.
Right. Lower F2006BP means less stimulation of glycolysis and less inhibition of gluconeogenesis in the liver. The net effect. The liver makes more glucose and releases it, raising blood sugar. Insulin has the opposite effect, increasing F2006BP and promoting glucose use and storage.
And you also mentioned xylulose 5-phosphate acting as a signal earlier from that other pathway.
Yes, this is a really interesting link. Xylulose 5-phosphate is an intermediate in the pentose phosphate pathway. When its levels rise, indicating plenty of glucose is being processed through that route, it triggers a cascade that leads to the dephosphorylation of that PFK2-FB-PACE2 bifunctional enzyme.
Opposite of glucagon's effect.
Exactly. This increases F2006BP, stimulating glycolysis. But xylulose 5-phosphate also activates protein phosphatases that turn on transcription factors.
Like CREBP?
Precisely. CREBP, carbohydrate response element binding protein. This goes into the nucleus and turns on genes for enzymes involved in both glycolysis and fatty acid synthesis.
Wow. So it's basically saying, lots of sugar coming through, let's burn some and store the rest as fat.
That's a great way to put it. It's a key integrator coordinating carbohydrate and lipid metabolism.
And the final glycolytic enzyme, pyruvate kinase, is also regulated.
Yes, it's inhibited by signs of high-energy ATP, acetyl-CoA, long-chain fatty acids. Makes sense, right? If the cell has plenty of energy or alternative fuels, stop breaking down glucose at the end. The liver version also gets inactivated by phosphorylation in response to glucagon, again ensuring the liver conserves glucose or makes it when blood sugar is low.
And even the fate of pyruvate itself, heading towards gluconeogenesis or the citric acid cycle.
That's regulated too. Acetyl-CoA, the product of fatty acid breakdown, activates pyruvate carboxylase, the first step of gluconeogenesis, and inhibits pyruvate dehydrogenase, the step leading to the citric acid cycle. So if fats are being burned, pyruvate is steered towards making glucose rather than being burned itself.
So these are all quick controls, allosteric effects, phosphorylation. But you also mentioned longer-term regulation.
Yes, transcriptional control. This is about changing the actual amount of these key enzymes in the cell by altering gene expression. It's a slower response, taking hours or days. Hormones like insulin and glucagon and signals like CHREBP play major roles here, adjusting the cell's overall metabolic capacity based on long-term dietary state or needs.
Okay, we've seen glucose 6-phosphate is central. It can go into glycolysis for energy, or be stored as glycogen, or be used to make free glucose via gluconeogenesis in liver. But there's another important path, isn't there? The pentose phosphate pathway.
Exactly. Glucose 6-phosphate is truly at a hub. And the pentose phosphate pathway, sometimes called the phosphogluconate pathway, is a crucial alternative route.
What's its main purpose? It sounds different from just energy production.
It serves two primary vital functions, and energy production isn't the main goal. First, and probably most important in many tissues, is generating NADPH.
NADPH, not NADH.
Right, NADPH. A similar structure, but used for different things. While NADH primarily carries electrons to the electron transport chain for ATP synthesis, NADPH is the main electron donor for reductive biosynthesis.
Building things.
Exactly. Building fatty acids, cholesterol, steroid hormones, processes that happen a lot in the liver, adipose tissue, adrenal glands. NADPH is also absolutely critical for protecting cells against oxidative damage from reactive oxygen species and free radicals.
How does it protect cells?
It helps maintain a supply of reduced glutathione, which is a key cellular antioxidant. This is especially vital in cells exposed to high oxygen stress, like red blood cells, or tissues like the lens and cornea of the eye.
Okay, so NADPH for building and protection. What's the second function?
The second major product is pentose phosphates, specifically ribose phosphate.
Ribose.
Yeah.
Like in RNA and DNA.
Precisely. Ribose 5-phosphate is the essential precursor for synthesizing nucleotides, the building blocks of RNA and DNA, and nucleotide coenzymes like ATP, NADH, FADH2, and coenzyme A. This is especially important in rapidly dividing cells, bone marrow, skin, intestinal lining, and unfortunately, also tumors.
This pathway sounds particularly critical for red blood cells with that NADPH protective role. Is this related to G6PD deficiency?
Yes, absolutely. This is where it gets clinically very relevant. The very first enzyme in the pentose phosphate pathway is glucose 6-phosphate dehydrogenase, or G6PD.
Okay.
Genetic deficiencies in G6PD are actually the most common human enzyme deficiency, affecting around 400 million people worldwide, particularly prevalent in certain regions.
And what happens in those individuals?
Because G6PD is faulty, they can't produce enough NADPH, especially under conditions of oxidative stress. This leaves their red blood cells vulnerable. Certain drugs, like some antimalarials, herbicides, infections, or even eating fava beans can trigger a massive oxidative stress.
Fava beans. Is that the favism connection?
That's exactly the favism connection. The compounds in fava beans generate oxidants. In G6PD deficient individuals, the lack of NADPH means they can't neutralize these oxidants effectively, leading to damage and rupture, hemolysis of red blood cells, causing anemia. Pythagoras might have warned against fava beans for this very reason, though that's speculative.
Yeah. But isn't there also a link to malaria resistance?
Yes, and this is a classic example of natural selection. G6PD deficiency is most common in regions where malaria has historically been endemic: Africa, the Mediterranean, Asia.
Why?
Because the malaria parasite Plasmodium falciparum, which lives inside red blood cells, is itself quite sensitive to oxidative stress. The slightly higher background level of oxidative stress in G6PD deficient red blood cells makes it a less hospitable environment for the parasite.
So the deficiency provides some protection against severe malaria.
Exactly. It's a balancing act. The disadvantage of potential hemolysis under specific triggers is outweighed by the survival advantage against a deadly infectious disease in those regions.
So how does this pathway actually make the NADPH and the ribose 5-phosphate?
It operates in two phases. The first is the oxidative phase. Here, glucose 6-phosphate goes through two oxidation steps, catalyzed by G6PD and another enzyme. This produces 2 molecules of NADPH per glucose 6-phosphate and releases 1 carbon as CO2, yielding a 5-carbon sugar phosphate, ribulose 5-phosphate. These steps are essentially irreversible.
Okay, NADPH produced here. What happens next?
The ribulose 5-phosphate can then be isomerized to ribose 5-phosphate for nucleotide synthesis, or epimerized to xylulose 5-phosphate, that regulator we met earlier. What happens next depends on the cell's needs. This leads to the non-oxidative phase.
And what's the goal of this second phase?
If the cell primarily needs NADPH, but not so much ribose 5-phosphate, the non-oxidative phase recycles the pentose phosphates back into glycolytic intermediates, namely fructose 6-phosphate and glyceraldehyde 3-phosphate.
Recycles them. How?
Through a really complex and elegant series of carbon shuffling reactions. It involves enzymes called transketolase and transaldolase. Transketolase transfers 2 carbon units and, notably, requires TPP, that thiamine cofactor again. Transaldolase transfers 3 carbon units.
So it rearranges the sugars.
Exactly. It essentially converts, say, 6 molecules of 5-carbon sugars back into 5 molecules of 6-carbon sugars, glucose 6-phosphate equivalents, allowing the oxidative phase to run again and produce more NADPH without building up excess pentoses.
But if the cell needs lots of ribose 5-phosphate, for DNA replication maybe.
Then the pathway can basically stop after the oxidative phase, or even run intermediates from glycolysis backwards through the non-oxidative phase reactions to maximize ribose 5-phosphate production without making much NADPH. It's incredibly flexible.
So glucose 6-phosphate stands at this branch point, glycolysis for ATP, or pentose phosphate pathway for NADPH and ribose. How does the cell decide which way to send it? What controls the flow?
The primary control point is the first enzyme of the pentose phosphate pathway, G6PD. Its activity is strongly regulated by the availability of its substrate NADP plus EBC.
NADP plus A, the oxidized form.
Right. When the cell is actively using NADPH for biosynthesis or antioxidant defense, the levels of NADP plus rise. High NADP plus levels allosterically stimulate G6PD, increasing the flow of glucose 6-phosphate into the pentose phosphate pathway to generate more NADPH.
And when NADPH levels are high?
When NADPH demand slows down, NADPH levels rise, and NADP plus levels fall. Low NADP plus, or high NADPH, which competes for the enzyme, inhibits G6PD. This slows the pentose phosphate pathway, and more glucose 6-phosphate is then available to enter glycolysis for APP production.
So it's directly responsive to the cell's immediate need for NADP.
Oh, precisely. A very elegant supply and demand system controlling the fate of glucose 6-phosphate.
It really is amazing. Everything seems incredibly interconnected, regulated, and responsive, from a single sugar molecule to the energy balance of the entire organism. So, wrapping this up, what does this intricate dance of glucose metabolism mean for you, listening right now? We've journeyed through how your cells expertly manage sugar, breaking it down for quick energy via glycolysis,
Synthesizing it anew via gluconeogenesis when supplies are low,
and diverting it through the pentose phosphate pathway to generate vital building blocks for DNA and protective molecules like NADPH.
Yeah, if you connect this to the bigger picture, it just highlights the stunning adaptability and sheer efficiency of biological systems. Every enzyme, every pathway, every regulatory switch is finely tuned.
All working together to maintain balance, homeostasis, ensuring your body has the right fuel and the right materials at exactly the right time, whether you're running a marathon or just sitting thinking.
It's all happening constantly at the molecular level.
It truly is a metabolic masterpiece. These microscopic decisions being made constantly inside you. It makes you think, doesn't it? How your body is constantly adapting, making these molecular choices. What happens if just one tiny decision point goes wrong? What kind of unexpected ripple effects could that tiny error have throughout your entire system? Something to ponder. Well, that's all the time we have for this deep dive. Thank you so much for joining us on this incredible journey into the cellular world. We're really glad you're part of the Last Minute Lecture family.
It's been a real pleasure sharing these biochemical insights with you today.
Until next time, keep exploring, keep asking questions, and stay curious.